Welcome everyone to this Studyclix Masterclass. We are live streaming directly to you this evening. Thanks a million for joining us. We're delighted to have you. You are in the right place. It's the right time. We're getting ready to have our Leaving Cert Biology Masterclass with Liam. So settle in, get comfortable, grab a paper, a pen, something to take notes, because we have some really useful tips to share with you tonight. Just to kick off, by introducing myself, my name is Amanda. I work for Studyclix as a schools engagement manager, which means I mostly work with the schools who have set up student access to Studyclix through the school. And if you're not already aware, if you've been kind of living under a rock for most of secondary school, Studyclix is a website that tries to make exams a little bit easier for you by providing resources that are designed to improve your knowledge around topics, but also your overall exam confidence and technique. We do this by categorizing past state and mock exam questions by topic, along with extra resources such as examiner annotated sample answers, solution videos, flashcards, and topic-specific quizzes. These are, of course, super useful for your big exams like the junior and the Leaving Cert, but also smaller class tests and in-house assessments that pop up throughout the year. Setting up an account is super easy. You can just visit studyclix.ie and follow the steps. The basic account is free, which gives you a taster of all of the features I mentioned just there, but you might like to avail of one of our paid plans. All info about that type of stuff is on the website. You can check it out after if you don't know already. And we're delighted tonight, though, to host this free masterclass for any Leaving Cert Biology students. This evening's session is specifically with the 2026 Leaving Cert in mind. So if you're currently in 5th year or tuning in just because you're a 3rd year who's thinking ahead about taking Leaving Cert Biology, feel free to stick around. Some of the tips and advice are transferable, but please bear in mind that the session itself isn't fully geared towards the new course or the 2027 exam. So just take what we say with a pinch of salt as a result. However, if you are currently in 6th year and taking biology, we're running this session especially for you, ahead of your mocks and your state exams next year. I'm delighted to be joined by Liam Hennelly this evening, who will share his top tips for improving your biology grade. Liam is a face and a voice you may already recognise from your use of Studyclix, because he's worked with us previously on things like the examiner, annotated sample answers, and Biology Boost. He's a state examiner himself and a brilliant teacher who has a lot of insight about how this subject is assessed, and he's ready to share that with you. Just before I hand you over to Liam, a reminder that this is being recorded and is available for you to watch back straight away after on YouTube, and also you can check out Studyclix itself to watch back the recording and from tomorrow onwards. And we've also published an article about this topic, and with kind of Liam's tips summarized. You'll find this on the biology subject page in Studyclix. And feel free to pop any questions you have into the chat, and we'll leave time at the end to answer some of them, which is when you'll hear from me again. Thanks again for tuning in, and I'll hand you over to Liam now.
Thanks very much for that, Amanda. Welcome, everybody. I'm going to take you through my kind of tips and tricks for Leaving Cert 2026. Very much will be, I suppose, end of an era. This course was first examined in the year 2004, and it's going to come to an end in June 2026. So we'll start off with the basics. Like the main thing that you have to know is the layout of the syllabus, and maybe not to break it down into topics, but to break it down into units, because that's how the exam is put together. And across the 400 marks of the exam, across the paper, there has to be an even split across all of the units. So Unit 1 is scientific method, characteristics of living things, biomolecules, which would include the structure of your food, micro and macronutrients, as well as the structure of DNA. And the big topic there would also be ecology. In Unit 2, you have cell structure, cell metabolism, which would include the big biochemistry topics like respiration and photosynthesis, cell continuity, which would mean mitosis and meiosis, and genetics then, which would cover crosses, evolution, genetic engineering, things like that. And then we move on to Unit 3, which would be human and plant biology, would also then incorporate fungi, bacteria, and protista, all under the various headings of structure, transport, response, and reproduction. So if you're sitting here this evening as a sixth-year student and you're wondering where to start, the first thing that I will be saying to you is to organize your resources. And so what I mean by that is, like, do you know where your notes are? Are they in a physical file? So what I would say is create a single folder. It can be physical, it can be digital, with labeled sections for the units 1, 2, 3, as I have outlined there. Use flashcards for definitions, for your key processes, and for your experiments. And then try to use mind maps to connect different themes. So if you start off and you write down the word DNA, can you then go off in tangent and talk about replication and protein synthesis and mutations? Try to fill in the gaps in your knowledge quickly. You can look up short videos and then make focus notes and then just try to study using targeted questions. This will usually be the best way. My main piece of advice here on how to study effectively is to study in the same way you would do the exam itself. So study like the exam. So what do I mean by that? What I mean is you have to assess where you are, right? If you're not assessing, you're guessing. So where are you on a topic? So I would advise going into Studyclix, doing an exam question, giving yourself the allocated time, and then going back and marking yourself by revealing the marking scheme and giving yourself a score out of however many marks that question is for. When you're practicing these, Section A questions should take you approximately 6 minutes. A Section B or experiment question should take 15 minutes. And a Section C long question should take approximately 22 minutes. So I'd be saying to you, make sure you have those numbers in your mind and you have a timer and you're able to give yourself that amount of time and then mark yourself. After you've gone through your answers, mark using the official marking scheme, which is embedded into Studyclix, identify your errors, and then go back and study these parts by using your flashcards or your mind maps. Then retake the question and compare the score. And hopefully then your score will improve. So repeat until your score does improve, you're up to the level you want to be at, hopefully H1 standard, and then that your timing for these questions is comfortable. What I would now go on and mention is how Studyclix Boost can accelerate this learning cycle, because it combines bite-sized videos, notes, flashcards, and questions into an actual learning pathway. And access to this will cost you $74.95 for a whole year. So for the price of essentially a grind in many, many other subjects, you can have access to all of these notes on Studyclix. And I'm just going to go through that with you now. So if I go into Studyclix here and I click on the topic, so let's say we're studying digestive system, and I've identified that I want to practice my digestive system. So this is a Section A question. You can see it here, question 6 from the year 2025. I put 6 minutes on the clock and I will complete this question. I would then go down here and go to show the marking scheme. And let's just say, for argument's sake, that for part B, I didn't know that the answer was Paracelsus. Okay, and I don't really remember what that definition is. So if I have access to Boost, I can go in here, I can click on a video like this. Long topic, and students do find that there is a lot of information to learn. And so if you haven't seen this before, what you can see is that you actually have a learning pathway. It tells you what you need to know. It goes through the videos, and as you scroll down, you can go into all the various topics here of the stages of digestion, the need for digestive system, and so on. So if I click here on mechanical digestion, I can go into the next video, talks about the teeth, and goes into all the different flashcards and learning definitions. And here's that definition of Paracelsus that maybe I didn't know on that last question, and here it is. So I could take that, make my own flashcard, and then I could go back and repeat that question, and then I could be up to the standard that I need to be at. Okay, so I'm just going to go back into OneNote here and just continue on going through this with you. So some basics. So the Leaving Cert has specific exam instructions, and you have to write your exam number and your day and month of birth in the box in the front cover. You need to write your answers to all parts of the exam into the answer book. And the way that you lay out your work here is really important. And I'd be suggesting that you put in the parts in the left-hand margin and you skip lines between parts. You want to make it really, really legible. there's certain boxes in the pages, and if you write outside the boxes, you may not get that work to the examiner because all the work is actually scanned. So your Leaving Cert Biology paper will be sent down to Athlone. Somebody will scan every single page that you write on, and that will be sent off to an examiner electronically. So if you don't write inside the boxes, some of your information may not make it to your examiner. When you're writing, you should only write in blue or black pen. You may use pencil for sketches, graphs, and diagrams only, and I would use a red pen for labeling lines. In the same way that there are 3 units on the course, there are 3 sections in the exam. You've got section A, B, and C. The questions for section C are supplied separately, but the answers must be written in your answer book. And it's recommended that you spend no more than 30 minutes in section A, another 30 minutes in section B, and then this would leave you with 2 hours for section C. So what I'm going to go through with you now is the exam instructions, how each section is laid out, and a bit of advice or kind of an expert tip on what I would do in each situation. So section A is just known as the short questions, and it's worth 100 marks. There are 7 questions and you're expected to do 5. There will be at least 2 questions from each unit of the course in section A. It's fairly predictable what comes up each year. There are a few curveballs thrown in every now and again, but almost every single year, question 1 is food, biomolecules, and the food tests. Question 2 is usually from the ecology section of the course, and so they are the 2 questions from unit 1. Questions 3 and 4 will be from unit 2. 5 and 6 will be from unit 3, and then question 7 could be any question, or it could be from any of those 3 units. Now, my advice here is that the short questions do not take a long time. I said earlier on that you should give yourself 6 minutes, but very often you might get done in 3 or 4 minutes. And so my advice here will be to do an extra question, because if you do, your best 5 will count. Another thing you need to be aware of is that negative marking applies. So if a question asks you for 1 answer and you write down 2 answers and 1 is right and 1 is wrong, you will end up with 0. Okay, so if they ask you for 1 answer, try to just give 1 answer. In a maths test, if the question was a basic one like what's 2 and 2, if the answer is 4, you wouldn't also write down 5. Okay, so try not to write down more answers than they ask for. Now, section B is based on the mandatory experiments, and there are 24 on the Leaving Cert syllabus for biology. These are also roughly split across the 3 units of unit 1, 2, and 3. And the big tip here that I would kind of try and share with you this evening is, of the 24 experiments, the questions that will be asked for question 8 and question 9, the experiments have to come from the list here. Okay, so question 8 has to be from this list, which includes the microscope, food tests, and the ecology experiments, along with the DNA isolation experiment. Question 9 has to be from all of the enzyme experiments and the effect of CO2 and light intensity on photosynthesis, osmosis, and alcohol. So you have an awful lot of experiments here on enzymes that are linked together. So if you study enzymes, you can kind of box off 5 of those experiments. I would be suggesting that you will not do question 10 next June, because all of these experiments in this list are from very separate and disparate parts of the course. So my big advice to you would be, of the 24 experiments, if you study the 9 for question 8 and the 8 for question 9, you really only have to study for 17 of these experiments, and you're guaranteed to get 2 of those questions. So that way you can save yourself a bit of time on studying and not do the experiments there on the right-hand side. So again, in section B, any 2 questions from the 3, each question is worth 30 marks. Question 8, as I said, is usually from unit 1. Question 9 will usually be from unit 2. And question 10 will usually be from unit 3. Another thing just to be aware of is that in these questions, part A is usually on theory and is worth 6 marks. And then part B will be worth 24 marks and is taken from the experiments themselves. So on to section C now. Since COVID, the Leaving Cert Biology paper has gone from 15 questions, do 11 in total, to 17 questions, do 11. And so in section C, you now have a huge amount of choice in that it's 7 questions, do 4. So the breakdown is as follows. Question 11 or 12 is usually a full question on ecology. Question 13 is usually on DNA. This is like DNA structure, replication, protein synthesis, and or genetics or genetic engineering. Question 14 is usually on biochemistry. And what I mean by that is it's usually a full question on either respiration or photosynthesis. Or in recent years, what they've done is they've halved it and they've given 30 marks for photosynthesis and another 30 for respiration. Question 15 will usually be a full question on one of the human biology organ systems, or it could be on plant systems. And then questions 16 and 17 are known as the half questions, because in these two questions, there are 4 parts, parts A, B, C, and D. Each is worth 30, and you have to complete 2. Now, if you complete 3 or even all 4 parts, A, B, C, and D, your best 2 will count. And topics that tend to come up in these half questions, so questions 16 and 17, are bacteria, fungi, viruses, evolution, plant reproduction, senses, protista, and cell transport, so things like osmosis. Now, my kind of advice or tips here: ensure that you answer section C in the separate answer book. Be really careful with how you lay out your answers, and I'll give a couple of examples of that later on. Use the left-hand margin to write in the question part, and try to skip a line between parts. Make your work really neat and legible for the examiner. Label diagrams really carefully, and know that it doesn't matter how good your diagram is, if it doesn't have a label, it cannot get marks at all. In in section C, I personally would focus on unit 2. So if you know DNA, genetics, and biochemistry, you're pretty much guaranteed 2 long questions. And I would then be advising my students to look at questions 16 and 17 and picking my top 2 parts out of the 4. So again, you have a lot of choice here. I would also encourage students to do a 5th long question on the day of the exam, and maybe focus on ecology for this extra question. Ecology tends to make up 1 short question and 1 long question every year. So this means that if you know your ecology really well, it can be 80 marks out of 400. And so it is essentially the only absolute banker topic. So 1 short question, 1 long question, 80 marks out of the 400. Another thing that I would encourage you to do on the day of the biology exam is to stay there for the full 3 hours. Hopefully you will not have to do that exam ever again. And I feel you should not leave early. You should spend the full 3 hours. And if you can, do an extra question in section A and in section C. I now want to move on and explain the concept of negative marking, another thing called cancelled answers and lists. So in section A, negative marking does apply if you give extra answers. So if we take a look at this example, the walls of xylem vessels are reinforced with, and the marking scheme says that the answer is lignin. But in the sample answer here, what you can see is that the candidate, right, the student, has given 2 answers, chitin and lignin. 1 answer is right and 1 answer is wrong. Therefore, this student scores 4 minus 4, so gets 0 marks. The next sample answer would be lignin, but the word lignin has been crossed out. The answer, which is correct, was cancelled by the candidate, but there's no other answer. So the examiner will always try and do their best on the part or on the behalf of the student. So in this case, the student would get the 4 marks. And in the last sample answer on this question, lignin is written down, chitin is also written down, but it is crossed out. So there is a surplus answer, but this time, as it is crossed out, the marks can be given. So the 4 marks will be awarded. Now, if we look here at this question, it says, name the 4 elements that are always present in protein. And the answers are carbon, hydrogen, oxygen, and nitrogen. In this sample answer, the answers given are carbon, hydrogen, oxygen, nitrogen, and calcium. There is a surplus answer, which is incorrect. So this effectively cancels out one of the right answers. So for the 12 marks that could be awarded, this student is now only going to get 9. So it's kind of like 4 3s minus a 3 leaves this student with 9 marks. The next sample answer is carbon, hydrogen, oxygen, and calcium. This time there are 3 right and 1 wrong, but because 4 answers were asked for, this student is going to get 3 3s for 9, and they are not penalized. The next sample answer gives us carbon, hydrogen, oxygen, calcium, and aluminium. An extra answer is given. 2 of the 5 are incorrect. So this student is only going to get 2 3s because the extra answer that is incorrect is going to cancel out one of the answers that was correct. And finally, on this one, we can see the same again, except one of them is now being crossed out. So there are 4 answers given. One of the surplus answers is incorrect, but the other surplus answer has been crossed out. So this student is going to get 3 3s for 9 out of 12 total. So that is the state of play for section A. And what I would say is look for the command words in the question. And if it says clearly to name 4 elements, we'll make sure that you just answer 4. There's no need for you to write any more than that. We move on now to section C, and we look at the idea that there is no negative marking here, but there is something called a list system. And so in section C, the long questions, if we look at this sample, it says state 3 functions of the cerebrum. And the marking scheme gives a whole host of answers here, okay, because there's so many functions of the cerebrum. This student has given a sample answer which includes memory, learning, and speech. There are 3 correct answers, so this time the student gets 3 3s and gets all 9 marks. In the next sample answer that we look at, we see memory, conscious thought, learning, and speech. The candidate, as you can see, has given us 4 answers. And if I'm marking this as an examiner, because I've only been asked for 3 functions, I am going to mark the first 3. So I'm going to say correct, incorrect, correct, and then I stop marking. Even though the 4th answer is correct, I can't award the marks because the question clearly said to state 3 functions of the cerebrum. So this student will actually only get 6 marks out of the 9 going. So what I would like to focus on with you now and maybe go through with you is a guide on how to approach some questions and also how to break down a long question. So here's a short question that you will find in section A. It's nearly always going to be question 1. Name the 3 chemical elements that are present in all carbohydrates. So it does say name, so I would be writing them down as opposed to just putting in the chemical element symbol. Carbohydrate literally means carbon, water. So it's carbon, hydrogen, and oxygen. And so that's what you should write here in the answer. Okay, so carbon, hydrogen, and oxygen. This question, the next one, part B, comes up so much. Give the general formula for carbohydrates. A lot of students here are going to make the mistake of writing down the formula for glucose. So this is not asking you to write down C6H12O6. This is asking you to write down CX bracket H2O Y outside the bracket. And you should be aware then that the ratio, okay, of hydrogen to oxygen in water is 2 to 1. Next question, name the smallest unit of a carbohydrate. And again, they're not looking here for the word glucose. They're looking for you to be aware of the phrase mono, meaning 1, and saccharide, meaning sugar unit. So the smallest sugar unit is a monosaccharide. Name the type of carbohydrate formed when many of the smallest units of a carbohydrate bond together. This really is asking, do you know that the word poly means many? So the answer to this question is polysaccharide. Give 1 structural role of carbohydrates in living organisms. And you should be thinking about plants here, and you should be kind of thinking of either cellulose, which is found in plant cell walls, and it does ask for 1, so that is an answer. I'm also just going to put a forward slash, and I'm going to put in the word chitin, which would be in fungal cell walls, because it does say living organisms as opposed to just plants. So there's 2 possible answers, and you could even have also said lignin. Carbohydrates are an important component of the diet. State 1 dietary source of carbohydrates. So again, there's so many answers here that a student could write down, but if you were to write down pasta here, or rice, or bread, or potatoes, you would be correct. Okay, a section C, or a long question, and how to approach it and how to break it down. So what you should notice in the long questions is that they're broken into 3 parts. So you have A and B and C. And they're always kind of broken up into 9 and 27 and 24. And generally speaking, okay, the marks are given in allocations of 3 marks. So if we look here at part A, it says, in relation to microorganisms such as bacteria and fungi, describe their distribution in nature. And what they're asking you here is, are you aware that they are basically found everywhere in most places all around the world? And that answer will be worth 3. And then, you know, you can kind of see here, you can see here that it says explain the terms asepsis and sterility as applied to microorganisms. And they are italicized, so they're asking you to define these words, and each of those would be worth 3 marks. Okay, so the next thing that we need to move on to then is drawing diagrams. And what I'm going to do here is I'm going to take this question. So the question says, draw the structure of a bacterial cell and label any 3 parts. So what I've done here is I've taken a page from the Leaving Cert Section C answer book, and I've put it in here into OneNote. And I'm on part B, I. So let's just say this was question 12. And it does always say start each question on a new page, just to be clear on that. I'm now working on part B, and I'm working on the first part. And the question is, draw a bacterial cell and fully label it. So again, you cannot get any marks for a diagram unless it is labeled. So I'm just going to really quickly here sketch a bacterium. This could be a rod-shaped bacterium called a bacillus. You need to remember that bacteria do not have a nucleus, so I'm just going to draw the piece of DNA here like that. I'm going to draw in a plasmid here. I'm going to put in a couple of ribosomes, and because it's a bacterial cell, I might just put in a couple of little flagella like that. So at the moment, just so you're really clear on this, this is getting 0 marks. And the reason it's getting 0 marks is there's no label. So at the moment it's D0 and it's L000. So the way this question is marked, it's 6 marks for the diagram, made up of 3 for the diagram itself and 3 by 1 for the labels. If I even do this, and I put in a labeling line and I write down cell wall. Now this student moves from getting 0 out of 6 to getting 3 for the diagram and 1 for the label. And if I just want to box this off and make sure I get all the marks, I can now add in a couple of more labels. I can say that that's a plasmid, which is a circular extra piece of DNA. I can say that that is a ribosome. You need to remember that bacteria have ribosomes because they need to make their own proteins. That could be the cytoplasm. This could be the DNA. And I would say to try and put in as many labels as possible. That is the flagellum, or the tail. Bacteria use flagella for motility or for moving. And now this student is up to the max, which is 6 out of 6. So if we go back up and look at the rest of the question and just try to break it down a little bit more. So a phrase that I want students to be aware of is the phrase command words. So when you're looking at the question, you need to be really, really conscious of what the command words are. So in the first example there, it was the word draw. And then the next question says, are bacteria prokaryotic or eukaryotic? And then this phrase that has come through from junior cycle science, justify your answer, means, are bacteria prokaryotic or eukaryotic? You give your answer, which would be prokaryotic, and then justify your answer basically means give a reason. Bacteria reproduce asexually. Name and describe this process. So the command word here, the first one, name, write one thing. So the answer is binary fission. Then the next command word or phrase is describe. So here you need to write in a number of bullet points. So binary fission, you'd probably be looking at 3 to 4 bullet points and a couple of diagrams. So just get used to saying to yourself, okay, what are the command words? What am I being asked exactly in this question? And then the next one is in italics. So it's a definition. What does pathogenic mean? Disease causing. And then antibiotics are used to treat pathogenic bacterial infections, but they're often misused. Give a possible effect of the misuse of antibiotics. And the answer here would be that they build up resistance and that eventually some antibiotics can become useless. So it is important that you're able to break down the 24 marks here or the 27 marks here and work out how much to write. So if I do a version of this here, it says, I'm going to skip the first part because it says the command word explain. So when I'm breaking these down with my students in class, I always go for the easy command words first and then work away at trying to work out how many marks are going for the longer pieces. So name the 2 biochemical components. Now that is protein and DNA or RNA. So there'll be 3 marks for each of them. So the whole thing is worth 24, and we're up to 6 out of 24 so far. It then says, give one example of a harmful virus and one way in which viruses can be beneficial. There will be 3 marks for each of them. So now that's 12 out of 24. And then it says, explain the difficulty in describing viruses as living organisms. This could be a sentence. This could be, okay, viruses do not demonstrate all of the characteristics of living things. They cannot reproduce. They can only replicate. So that will usually only get 3 marks. And so now we're up to 15 out of 24. And what that means is that this part 3, because of that command word describe, is going to be worth 9 marks in total. And so you will have to write a minimum of 3 bullet points, or combine 3 little bullet points with 3 diagrams to get the full marks here. And so that's how I would kind of start practicing when I'm doing my exam questions on my own, when I'm studying for my house exams, for my mocks, for the Leaving Cert itself. Start breaking these questions down, be very conscious of the command words, and be very conscious of all the other things that I've gone through in the webinar so far. Okay, Amanda, I've gone through all my material there. If there are any questions after coming in.
Brilliant. Thanks so much, Liam. Yeah, just a reminder that you can use the chat there, guys, if you want to ask Liam any questions. We have a few in already. I'll give you this one. Is there negative marking on diagrams, Liam?
It depends. Is it going to be a diagram in section A, or is it going to be a diagram in section C? If you label a part of a diagram in section A and it is wrong, negative marking can apply. And if you label a diagram, or if you draw an incorrect diagram in section C, it will just be marked incorrect. So it does apply in section A, but not in section C. If it's wrong, it'll be marked wrong. Okay,
perfect. Good stuff. Hope that clears that one up. Another one here about drawing diagrams. When describing, can I just draw the process? Will I get marks for drawing?
You will get marks for drawing, definitely. You may not get all the marks. And what you will often see in a question is, it might say, describe using a label diagram. And if you only give the label diagram and there is no description, then you cannot get all of the marks. What I would say, and my advice to my own students always would be, definitely draw, diagrams. If it's something like explain viral replication and you have your diagrams for the 3 or 4 stages, I would write a bullet point, a really short little bullet point to accompany each diagram. That old phrase of a picture paints 1,000 words is absolutely true, especially in biology. But back it up with a point. Like if you're in English class and you're kind of quoting something, or you're writing about a text, you will always back it up with a quote. So I would say in biology, draw a diagram by all means, but back it up with a sentence.
Very good. I think that's a fair enough tip. Not a really good question here. Just generally, how sensitive do examiners tend to be about spelling in these scenarios, especially for the really complicated words?
Yeah. So I guess there's a few things to be aware of here. Okay. Number one, some students will have a spelling waiver, and in that case, as long as the word could be made out phonetically, it will be fine. If a student doesn't have a spelling waiver, and if generally speaking the word can be made out phonetically, there should be no problem. But there are some occasions when they ask for certain definitions. So for example, if they said, what does ATP stand for? You would have to say adenosine triphosphate, and it would have to be spelled correctly. And if it was NADP or NADH, you would have to get it completely right. It would be nicotinamide adenosine dinucleotide. And I have seen them being very harsh, but in the main, unless it's a really, really particular question, they won't be too harsh. But I guess the answer to that question is, it depends. It depends on the circumstance of the student, and it depends on the question that was asked.
Yeah, that's fair enough. Thanks, Liam. This is kind of the magic question, I suppose. What is a study method you swear by? If you can pull one out of your head.
Yeah, I wouldn't have just one. I think you have to have multiple approaches. As I outlined earlier, my recommendation, and what used to work for me as a student, was testing myself by doing past questions and figuring out what I don't know. So if I do a question and I get 80%, then I know I need to study the 20% that I didn't know. What I would now say in this lovely modern era that we're in is that you guys have so much information at your fingertips that you need to try and make use of dead time. So if you're getting a lift into school, or if you're on a bus going home from school, you know, you don't necessarily always have to be scrolling social media. You could be finding YouTube channels. You could be on Studyclix watching videos. And there's so many different ways that you can actually access content nowadays that maybe I couldn't, 25 years ago when I was doing the Leaving Cert. So just be aware of that. Be aware of the power of the machine in your pocket, and how much information, how much content you can access. There are podcasts on Leaving Cert Biology. There are YouTube videos. So if you have some dead time, try to use it. Try to fill it productively, and maybe not always with scrolling on the social media websites.
Yeah, easier said than done, I suppose. But
Absolutely.
yeah, certainly studying isn't always just sitting down, reading your notes or reading the book. It's trying to think of more tolerable ways to get the information in, especially at times where maybe you don't want
and I suppose if I nearly flipped the question, a study method that I would bin is that, I would never recommend that a student take out a biology book and just read an entire chapter from cover to cover. That's obviously fine for English or for history, but I just find that doesn't work for biology. So if I could give one bit of advice, do not really sit and read a whole chapter back to back. You need to have that pen in your hand. You need to be drawing diagrams. You need to be asking yourself, do I know this definition? Can I write it out?
Yeah, exactly. And I think a really useful thing on the go as well, just a shameless plug for Studyclix while I sit here wearing a Studyclix t-shirt, is the quizzes, especially in biology, are fantastic for that quick fire round. Click into the topic that you know you've a test in that day while you're on the way to school. Studyclix will give you 10 questions that'll test you on that topic, multiple choice, freehand text. The best way to kind of just understand what you don't know is the key for studying. So that's a couple of tips there. Hopefully we helped you with that one. Kind of staying on track for like study methods, study tips. Do you have any kind of tips for that night before type of stuff? Or how would you approach that kind of mindset? The most productive thing to do the night before?
Yeah, if it's a full mock and the whole course is wide open, and maybe you're not fully finished the whole course, I would come back to ecology. And I would say, if you know your ecology, that's going to be 80 out of 400 marks. You can go through there. In a typical Leaving Cert paper, there will be 7 or 8 ecology definitions asked, and there are 42 ecology definitions on the syllabus. So my last minute hack for a student would be know your ecology definitions, and maybe know your carbon and nitrogen cycles.
Mm-hmm. Yeah, the cycles, I remember those ones as well. It's all coming back to me now. It's not that long since my Leaving Cert, but yeah, it's all coming back. Some stuff here just specifically about the sections. You give a really good comprehensive overview of what to expect from each section, but this one, come back to section B. If you only do one of the questions in B, what would happen?
Yeah, if you only answer one question in section B, then you are essentially marked out of 370, and you're down 30 marks straight away, and it means you then have to get 360 out of 370 to get a H1, or you have to get 320 out of 370 to get a H2. So don't do that under any circumstances. You have to try and do 2 out of 3 in section B, and hopefully by now being able to avoid question 10 and being able to say, okay, I only need to study 17 of the 24 questions, that should be helpful.
Great. Yeah, good advice. Going back to the cycles point we just raised there, just a question I spotted. Was on ecology and the cycle, what systems would you recommend knowing off well? Is there kind of a reliable bunch?
Yeah, this is a tough one because, as humans, as people, we're obviously very interested in studying ourselves. Like, I think everyone's favorite topic to study is a various human system, be it digestion or reproduction or whatever the case may be. You are drawn to things that you like. And my take on it would be that you cannot really predict what human system or what plant system is going to come up in this paper. It varies so much. So you have to kind of go at the things that are that bit more predictable. So for me, when it comes to systems like that, I would be looking at photosynthesis, respiration, the Krebs cycle, what's sometimes known as the Calvin-Benson cycle, the light-independent reaction. They come up on the paper with way more frequency than digestion or human reproduction. So if you do, and there is an analysis done by Studyclix on how many times these topics come up, and you can go on Studyclix and have a look at that, and you'll see the incidence. Biochemistry, respiration, photosynthesis, pretty much every single year, whereas digestion might have only come up 6 or 7 times in the last 22 years. So personally, I always push my students. I actually ask them to maybe move away from human biology. They don't like it, obviously, but when the results start improving, when they get a better result in the mock by following that technique, and then that's replicated in the Leaving Cert, they kind of see the reason why.
Yeah, nice, very good. I've had a question come in here about the difference between higher and ordinary level in your view, and maybe some advice for somebody who might consider dropping down or is in limbo about whether they should stick with higher. It kind of all feeds in.
It is detail, exactly. Everything that's on the ordinary level course is on the higher level course. And then the higher level course just has extra content. I've marked both levels. I've marked Leaving Cert ordinary level and I've marked Leaving Cert higher level. And sometimes, you will, like, I would say that O1s in ordinary level biology, they should be rare. Okay, and the reason that an O1 in a subject should be rare is because that student is more than likely well capable of maybe getting a H5 or a H4 at higher, and could possibly go up even another grade and get more points in higher than they would in ordinary. I wouldn't necessarily like to be quoted on me saying this to too many people, but if you were someone who's capable of an O1 or an O2, I think you should be doing higher level. And I would be kind of sticking with higher level in biology until after your mocks. And even if you were in the mocks, if you were scoring 33, 34, 35%, I would still be encouraging that student to take higher, because you would have 3, 4, 5 months to finish the course, to do your revision. And you do have to remember this PMA, this post-marking adjustment. Last year, I think it was 6 or 7%. This year, it was 4 or 5%. So like if you were a student and you got 38 and they added 4%, well, now you're on 42%. Now you're on a H6 and you're getting 46 points. So yeah, it is the difference is simply the detail, and obviously there's a lot more detail in higher. But I would be saying that an O1 standard student could easily do quite well, get a H4, maybe even on a good day get a H3 in higher level.
Yeah, I think that's really good advice. Like, it does vary per subject, I suppose, in terms of when to stick around in higher and ordinary. But I think biology is a great example of, it's the same course, it's the same information as your higher level peers. You might as well kind of.
Yeah, and I suppose even on that, just a bit of insight, like biology is the biggest option subject in the country. I think this year there was something like 33,000 students did biology across the country. And of that, I think there was 29,000 students did it at higher level. So it is the subject in the country taken at higher level. No other subject comes close. So biology at higher level is the most frequently taken higher level subject.
Yeah, very good. I think we will leave it there for tonight, folks. Thank you very much for your questions and stuff. I hope you found the session useful. As I said at the beginning, it is available to watch back immediately on YouTube after the stream. So if you missed the start or you'd like to go back over any of Liam's sections, you can do so immediately. As I also mentioned at the beginning, just in case you weren't around for it, we have a guide available on Studyclix written by Liam that kind of summarizes a lot of the points he made tonight. You'll find that on the biology subject page in Studyclix. Thank you guys for your time and attention, and a huge thank you for Liam for sharing his insights. And yeah, have a good evening, everyone. Thanks for joining us, and take care.